The reality is if you're focused on making a mark, you're probably not going to. If you're focused on helping and working with other people and collaborating, I think you'll be successful. It was actually in my second job that I worked with Chad that, I've told this story multiple times, is he was like, hey, can you help us understand what you're doing and why you do it? Because, we get it, but we don't get it, right?
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe.
We've seen the pharmaceutical industry change tremendously, right? When when you and I got into this business, it was really a small molecule business, or in your case, you were actually working with the PTH, which is more of a biologic. But we've seen it evolve significantly, which means different types of data, biomarker data, et cetera. Can you tell me? I'll say a pharmacokineticist or a pharmacometrician's view on how your role and responsibility and the role and responsibility of that particular piece in the industry has changed over time.
Yeah, it's changed a ton. You're absolutely right. Even though it was a small molecule world, I was working on a large molecule, a biologic, but everything's a biologic now. You have antibodies and antibody drug conjugates, and it's rare that it's a small molecule under development.
Yeah,
I think for me the biggest change that I have seen is trying to think of how to phrase this, but taking information you've learned before and making predictions of what you might see in the future from a biological perspective. So a lot of people call it model-informed drug development, which is kind of a mouthful. But the idea is you develop mathematical models and say, hey, we've seen this way before. What do we project is going to happen next? And when I first started, that was not done. We didn't do that regularly in the pharmacokinetic, pharmacodynamic space. But it should be done. And now we are doing it on a regular basis. So a lot of my work now, when I work with companies consulting, is they'll say, hey, we've run a phase 1 study. We want to run phase 2, but we're not sure what the right dose is that we want to use to hit this target or see this response. And so then I build a model off phase 1 data, and I project different phase 2 trial designs and doses and say, this is what I think it's going to be. And then we choose the dose to use in phase 2. Mm-hmm. When we started, it was, hey, what's the maximum tolerated dose we got in phase 1? Let's use that for phase 2 and see what we get, right? And you try to give as much drug as possible so you can see an effect as long as you're not hitting toxicity. And then maybe you back it down a little bit, right? Mm-hmm. That was drug development. And it was very crude. And now we're getting more refined with it. And I think that's this idea of the models helping describe what's going on. Our bodies are just amazing machines. Mm-hmm. They have a set of rules that we don't know or fully understand. But they follow those rules. And if we can help unlock those and understand those better, then I think we can get more predictive of what we're doing. There's a lot of ideas out there about virtual clinical trials, right? Mm-hmm. Can you essentially run a clinical trial in virtual patients and get results out of it to decide what you're going to do next? Can we eliminate some of the clinical trials we're running now that really aren't necessary? Right. Can we answer those with virtual things? So I think that's the shift that's happened in my discipline is applying these models to project what we expect to happen in the future.
Yeah. Oh, that's really cool. You almost touched on our guest from December, Tom Helikar. Yeah, Gregory's thinking
I was thinking the same thing. It's like how we just talked about this last month.
Yeah, he's a computational biologist, University of Nebraska, and you might find that really interesting to listen to some of what he's doing, or maybe, quite frankly, just dig into his literature and read some of it. But he's making a digital twin of the immunological system. So and super interesting guy as well. So he's going right where you're talking about. But we didn't even bring up the idea of doing virtual clinical trials. We were, but it absolutely makes sense. You can kind of control the variability a little bit as well in how you do that. So yeah.
Well, and you can do things like enrollment in clinical trials is one of the most costly parts of a trial. And especially when you're saying, hey, we need to get this distribution of participants. I know that one of the areas of concern from the FDA, specifically in the U.S., is making sure that we get a representative sample of the population. And oftentimes you'll get a couple different people. You'll get the sick people that are wealthy enough to understand that drug development's going on, so they're getting the best care from the highest paid doctors who are involved in clinical trials, so they're aware of them. So you get that population, or you might get a population that is individuals that have no other option, right? And so they're kind of forced into it. We do pay people to participate in clinical trials, and so they're doing it for the money. And there's a lot of people out there that are just hardworking people that never get exposed to clinical trials.
Yeah.
And how do we engage with those people? How do we get all those different people involved in clinical trials? One way is through virtual clinical trials, and we can evaluate it and understand, hey, are they really different? Yes or no? And if they are, then let's focus some energy to actually enrolling those different kinds of people. So I think it's exciting. I think hospitals could take advantage of this. I think they could take advantage of it in formularies. Hey, what's their population for a healthcare system? What is their population, and which are the drugs that would work best in their population? Yeah. Not taking drug data from the drug companies, but saying this is my group of patients in my healthcare system. Yeah. This is the best drug for us. Right?
I wonder if there's an opportunity, because all that data is out there and available, but it's all de-identified, right? Yeah. So you almost wonder if there's not an opportunity out there somehow to mine all that data, find the ideal candidates for clinical trials, and then sort of put it out there and say, hey, if you're patient, 42716, we're just going to send a blanket thing. Then we may have a trial that can help your very unusual condition because of XYZ. It's a tricky, slippery slope with privacy that's so important, and I do believe in that. With maybe we can find people and give them an opportunity to get on this trial.
Yeah, exactly. And it might be reaching out to the physicians that treat those individuals, right? Yeah. Rather than to the individuals directly. Yeah. But I just think it's interesting because we can access people that we didn't before, but we can also potentially not run some studies. How many bioequivalence trials do we need for things where could we run them virtually instead? Why waste our resources on clinical trials that aren't needed?
Interesting. And in your consulting work, Nathan, you mentioned a lot about the dosing. And I assume that's probably a very common hurdle in drug development is identifying what's the best dosage. Is that the most common hurdle that you come across, or are there other things that are also pretty common that you deal with?
Yeah, do you have the right dose is absolutely the biggest question. Do you have the right dose? And it's two perspectives. So you have the drug company saying, do I have the right dose to show efficacy to hit my primary endpoint to get an approvable drug? And then you have the regulatory agency on the other side saying, could you dose lower and maintain adequate efficacy to minimize potential toxicities? So the drug companies want to give the highest dose possible because it ensures they have efficacy.
Right.
But the upper limit is, okay, we don't want too much toxicity, so we need, something that's reasonable. And regulators, because their focus is public health, they say, what's the minimum effective dose? And so finding that balance is absolutely the right thing. I see drug companies looking at when they're in phase 2, picking the right dose. And then after phase 3, when they're submitting for approval, they're trying to justify their dose selection and counter any arguments from the regulatory agency. So I think this is absolutely the biggest topic. You probably heard of Project Optimus in the oncology space. So I think that's maybe 4 or 5 years old now. Yeah. But the whole idea behind Project Optimus is, hey, we need to do oncology development different. We have to choose the right dose, and we have to do dose finding in oncology, because they didn't. They would choose hey, what's the maximum tolerated dose, and let's just plow ahead with that. And hey, that works good for cytotoxic agents, but it doesn't really work for targeted therapies and immunosuppressants and all the other new cancer treatments that are coming out. And
it doesn't
so Project
really work
Optimus,
for the patients either necessarily,
No.
even in cytotoxic agents, right? Yeah.
Yeah. Because the right dose for me is probably not the right dose for you. And, nor is it the right dose for grandma who's 75 and has breast cancer now,
Yeah.
And so I think the real push in oncology is like, hey, let's choose the right dose. It's funny. Some people say is Project Optimus is telling people in oncology that you should do drug development like the rest of the pharmaceutical industry, because they did it differently. But the reality is dose selection and dose optimization it will continue to be the primary concern in drug development.
Greg, is it time again for new news?
I think it is time for new news.
New news. And new news.
All right, Nathan. So I was just reading this article. This was actually Fierce Pharma, which I think is what highly read by many people. So you might have seen this, but after 2 recent rejections, the FDA approved Mesoblast. So this is the pharma company's Mesoblast. After 2 rejections, FDA approves Mesoblast's first of its kind cell therapy. So it was interesting because the FDA advisory panel had voted 9-1 to recommend the approval of Mesoblast's graft versus host disease cell therapy. But then 4 years later, they had actually 2 rejections from the agency. So it seems that despite being recommended to approve, they had 2 rejections. But Mesoblast finally got endorsement from the FDA to have its first approved drug. So this drug Wednesday afternoon. This just happened, I think, last Wednesday afternoon, or it might have been just I have to look at the date. It might have just been this yesterday. But they signed off on Ryoncil, is the name of the drug. It's an off-the-shelf cell therapy to treat patients 2 months and older who have acute graft versus host disease and don't respond to steroids. So apparently in about half the population of these young children that get transplants, end up having graft versus host disease, and then the steroids just aren't a great treatment. So there are a few issues why they wanted more data on efficacy was part of the issue. They also asked for, and this stuck out to me, but Mesoblast adjusted its potency assays. So there are issues with the potency assay to not really demonstrate the identity, strength, quality, purity, and dosage form. So it's kind of that situation, maybe to summarize it, where they saw something, looked like it was working well. They wanted a little more evidence around efficacy, and then also they didn't have maybe quite the right assays to really demonstrate and give the FDA that full confidence. So this says of the roughly, this kind of the stats are here at the end of the article, but of roughly 10,000 patients who have undergone an allogeneic bone marrow transplant each year, about 1,500 are children. And so, half of them develop acute graft versus host disease. So one of the reason I picked up on this is I just actually recently been talking to a good friend in the industry who works in cell therapy, and he was saying, the troubles with cell therapy and there's all these different challenges in moving forward cell therapy. So that the initial promise, which we all know is still there on cell therapies, has been fraught with all these challenges of anything from complex manufacturing, high costs. It's hard from a regulatory standpoint, which Mesoblast clearly encountered. Scalability, how do you run it in the clinic? I mean, there's all reimbursement issues. So, anyways, I'll kind of stop and get your take on this situation and this area a little bit.
Yeah, cell therapy it's really interesting. I mean, the basic idea of, and I don't know the specific one, so I don't know if this is a CAR T type approach, but usually they extract cells from an individual, they go process them, ex vivo outside the body, and usually they're injecting some sort of transgene into them, and then they inject those same cells back into the patient, and supposedly it's supposed to restores some function to their immune system to help. And a lot of times it's cancer, blood cancer, I think was the first area that these were developed. But what I think is really interesting about this is it's not just about the dose that you give of these cells. It's about sort of the individual body's capacity to expand those cells. So if you kind of think about it, most of these are immune cells. And so the way they treat people is they'll draw some blood out, and they're manufacturing the blood cells on the side to create the dose for that patient. At the same time, the patient is going through a process to deplete all of their immune cells. And then what the hope is when they've depleted all their immune cells, they get injected back these modified cells. And then those cells are going to expand inside that patient's body. But as you can imagine, every patient probably has a different immune system capacity. Right. And maybe they call it a niche or a space or whatever it is. So what happens when, one patient has a really small space, right? It doesn't matter how many cells you give them. They have a small space. It's not going to do much. But you have another patient has a really large space. Well, then the number of cells is important. I think it's a really intriguing question, and it's one of those ones where I don't think we have the right answers. We're approving treatments, and I think that's why the FDA is having trouble with this, is they don't know what the basis for approval is necessarily. What is the right dose? How do you tell people what the right dose is? And there's a set of complex factors. This is where maybe if we go back to thinking about AI analysis, is we don't understand the mechanism or the factors involved. But maybe if we put the data together and ask some of these AI network models to build a model, maybe it can find, the right triggers and switches that say, hey, this is how you dose this patient. These are the factors that are important. But we have to collect all that data and put it in a pool together and analyze it. So I think it's kind of cool and interesting. It's very challenging, though, from drug development perspective.
Such a challenging space. And I respect so much the companies, the executives, the scientists, these folks that continue to press forward in these in the space it's so hard, right? There's so many things in the pharma industry that are so hard. So
I love the promise, as you both have mentioned, of cell therapies, but I'm curious. I don't know enough about the side effects, but based on what I just heard, I didn't know that the immune cells, white blood cells, are actually being really removed or reduced drastically so that it'll take in the new modified cells. Is that leaving people immunocompromised in the process if it doesn't quite work? Is that one of the big issues?
So, I mean, yes, they are immunocompromised, but the immune system restores those cells. The hope is that because you're injecting a bunch of these modified cells in, that those restore, those are the majority that are restored.
Right.
And so then you get some treatment benefit over a period of time, and then you might have to dose again. And so, I mean, being immunocompromised is one. There's a huge, from a bioanalysis perspective, Chad. How do you detect this stuff? These aren't small molecules. They're not antibodies. They're actually cells. And there's multiple methods that people have, but they all give different answers. And which is the right answer? I don't know. Right? It is really kind of crazy.
Yeah. Well, it's brought a whole revolution to bioanalysis, where now, you're all the bigger bioanalytical labs out there now have added flow cytometry, PCR, ELISpot, all these different technologies, doing so many more cytokines to monitor, because Greg, while you're depleting the immune system, at the same time, you got to charge it up so that the new cells go, and sometimes they go crazy, right? Like, Nathan said, there's a lot of variability. So sometimes you charge it back up. When you charge it up, it just, explodes with activity. I don't want to make it sound like you're exploding, but so you monitor these cytokines and other markers to see what's happening. You measure cell expansion with flow cytometry. So kind of going on and on. But it's such an exciting space that yesterday, when I was talking with my friend a little bit, and he was he was kind of down because things weren't going well in the company he's at. But at the same time, then I saw this article. I'm like, there is promise in this space. We got to keep moving this forward. So
Indeed.
So yeah, very, very exciting, very interesting. Yeah.
And it's great to see it also in pediatrics as well, which is probably more rare.
I was really
cell
surprised to
therapy.
see that.
Yeah. It's cool.
Yeah.
So Nathan, across everything that you've experienced and learned in your career and maybe some of the mentorship opportunities that you've had in the past, what advice would you give to aspiring scientists and or budding entrepreneurs in this space looking to make a mark in this industry?
Wow. That's a big question. Actually, the reality is if you're focused on making a mark, you're probably not going to. If you're focused on helping and working with other people and collaborating, I think you'll be successful.
Sure.
I'll be honest, my career, I started off focused on the science I did and my science, and I wanted to publish papers. That's what I learned from my academic lab, right? That was important. And it was actually in my second job that I worked with Chad that, I've told this story multiple times, is he was like, hey, can you help us understand what you're doing and why you do it? Because, we get it, but we don't get it, right? Yep. And I came in and we spent a lot of time back and forth, and I was trying to say, hey, this is what I do, and this is why when I ask this question or I need this information, this is why I need it, because this is how it affects me. And I realized that my success was going to be based on collaborating with other people and helping them understand what I'm doing and why I'm asking a question. And that's going to be more important, is kind of building those bridges and contacts with other disciplines. Yep. And so that's what I've done. And that's my career, is actually grown out of that. So bioanalysis, clinical development, medical directors, toxicologists. I work with tons of different people, and my ability to connect with them and say, well, this is why I need it and why it's important to me. Can you help me here? And they would explain, well, we can't do that because of X, Y, and Z. I'll be like, oh, I understand that now. And so if you want to be successful, I think you should, maybe seek first to understand, and then you'll be understood, kind of thing. If you actually are helping your colleagues and saying, I'm trying to do this, but I need some help here. Can you help me? You're going to get a lot further, and then you're going to leave a mark if you do that. Does that make sense, Greg? Or is that
it does?
Concept?
It does. And I think that's great. It's not exactly what I meant when I first said made a mark, and I can see why you went that way, but I think it brought up a very key important, that it's not about me, it's about we. And that's the only way you're going to be successful and grow a good career in life sciences is taking that collaborative approach, which is a perfect lead into the question from our last guest is, how do you measure success in life?
Oh, how do I measure success in life? I think it's happiness of the loved ones around me. So if we're happy, then that's success for me. I have my wife, I have a couple kids, my kids are married now, and there's nothing better for me than hanging out with them and being happy with them, and that's success. Everything else is sort of secondary. And actually, I think that goes into a little more about, like, what do you do in your career? I have people ask me, how do I do this? How do I do that? Find something you enjoy and then do it, right? The career that I'm doing now didn't exist when I was in graduate school. There was no such thing as a pharmacometrician when I was in graduate school. So I would guess that the same thing exists for most people that are in their careers today. In 15 or 20 years, what you're doing probably doesn't exist. So don't hyper-focus on, I need that job today. Figure out what you like doing. And for me, what I like doing is I like helping other people solve problems. That's what I actually like doing. And I think that's what I'm good at. So I have a discipline, pharmacometrics, fine. I use pharmacometrics to help solve problems for other people. But what I enjoy is helping them solve those problems.
That's brilliant. I love that.
I love that.
And I think that's a great message.
Yeah. Nathan, speaking of helping other people and helping them solve their problems, I am a fan of your podcast.
Oh,
and
thank you.
I've told so many people that, that I know, either I might hear him talking about PK or something. I'm like, you got to go listen to this guy, Nathan. You got to go listen to his podcast, because people that are working in the lab I run, I'm like, you got to listen, for the trainings, just go out there and listen to it. It's a nice length, easily digestible. I would say it was, at least from my viewpoint, like sort of like PK 101 or Clin Pharm 101. Now we might be into 201 now as I'm listening to it. I got to pay attention a little more. Let's put it that way. But that's the natural evolution. That's what it should do. So, so can you talk a little bit about your podcast for us and what your goal is with that, where that's going? It's great. And tell us what it's about, since I didn't do that.
Yeah. First of all, thank you. I appreciate it. It's, I was, after I left my last company and I started the software company, I'm like, what should I do? And I'm like, I used to have a blog that I posted stuff up to for many years. And that blog's no longer mine. I sold it to another company. And so I was like, I should do something new. And I was like, well, I'll try a podcast. And I have been amazed at how many people listen to it and give me similar feedback, Chad, of, this is great. I'm not in that discipline, but I appreciate you talking about this. Mm-hmm. And I'm stunned. I mean, the things that I talk about, I try to talk about, hey, these are some standard things that we do, and this is why we do them. Go back to what I was saying earlier about, building those bridges. This is why I do what I do. This is why we calculate this parameter, and I think this is important, and I think this is not important. Mm-hmm. And just talking about those concepts. And I think people are generally interested in learning about other disciplines. Like in bioanalysis, where does this data go? Why do they need it? What are they using it for? Right? Your science, but you don't know what people do with it downstream. Right. And I think that's why people enjoy it, is they get to hear something else. So the topic is clinical pharmacology. And so I talk about pharmacokinetics, I talk about pharmacodynamics, I talk about modeling, I talk about data analysis, I talk about bioanalysis, all the sort of things, as clinical trial design, all the things associated with clinical pharmacology. And the idea is not only to be a resource for people in the discipline, but adjacent people to listen and go, oh, I understand that now, or that makes sense, or I never knew that. And so I've been going for, about 18 months with the podcast, twice a month. And, nice. I got a new year coming up. I'm actually trying to figure out what I'm going to talk about next year. Am I going to change? But I've really enjoyed it because it's been a nice way to connect with people on not having to go to a meeting, not going to one-to-one. It's kind of fun to go to meetings and have people say, oh, I've listened to your podcast. I have no idea who they are, right? I don't know who listens. I just know there's 300 or 400 people that listen every time I put one out. Great. But again, awesome. I'm glad it's helpful. So you can find it on Spotify, on Apple Podcasts, wherever you listen to your podcast, just search clinical pharmacology. You'll find it. And I always take suggestions. So if you listen to a podcast, there's an email, a website. You can go there and send me, hey, talk about this. And I get a lot of my topics from people requesting information.
Well, Nathan, I would make a vote for what happens to our data after we send it to you. And I'm saying that as the bioanalytical guy, right? And you've talked about how you do PK, how all those. But I think that could be an idea, right? Like what sort of
what really happens to the data.
How's the sausage made? I send it to you. Why does it need to be in that format? And what do you do with it? And then where's it go on the back end? I think that might be interesting. It might get, from one podcaster to another. It might be something that pulls in a few other people if they see, bioanalytical. You might get a few more bioanalytical folks that are kind of like, maybe I need to get into that, and then it could tie back to a bunch of your earlier episodes. So
sounds like I have a January topic now. So thank you, Chad.
All right,
talk about that in January.
That'd be great. Okay.
Excellent. So Nathan, what's the best way for people to reach you? Is it your website?
Yeah. So to reach me about aplosanalytics.com. is the best way to kind of get in touch with me. You can also email me at nathan@aplosanalytics.com. And that Aplos is APLOS. You could also go to TeuscherSolutions.com. That's where my podcast is. You can listen at the website there. Like I said, you can search on Spotify and go on LinkedIn. If you find me on LinkedIn and send me a message, I'll respond on LinkedIn as well. So, that's one of the things I enjoy about the pharmaceutical industry is we're a small group. And in my opinion, we're a pretty friendly group. We're a pretty collaborative group. Doesn't matter what company you work for, we're all working on the same thing. We're trying to develop drugs that, reduce human suffering due to disease. And we all want the same thing. We all want people to feel better, be healthier, and enjoy their lives. And so I think we do collaborate really quite well. And, people might see the pharmaceutical industry as a, big behemoth that's, out to get people. But the reality is the scientists working in it, we're super collaborative, and we're great people, and I just love working with people. So yeah, happy to reach out and happy to make connections and help. And I really appreciate you guys having me on. So it was a lot of fun.
Excellent. I think it's our pleasure. Yeah. Thank you, Nathan. Really appreciate it.
Yeah, it was a lot of fun. Really enjoyed the conversation. I know we could probably go on for another hour. But I'll make sure all those links and so forth to how to reach you and your information will be in the show notes on YouTube and Spotify and all those areas as well. And thanks again so much for being here. We really enjoyed having you.
Yeah, thank you. Happy holidays, guys.
All right, thanks, Nathan.
Happy
holidays to you as well.
Yeah.
Thank you for listening. Please check out Nathan Teuscher's podcast, Clinical Pharmacology, on Apple, Spotify, and wherever you get your podcasts. Links are in the show notes, and we'll see you next time.